Before I get started, Dr. Chambers wanted to say a few things.
Yeah, so a lot of the things she just mentioned we've solved. We've been working on this project for over 5 years. We just received a NIAID grant for $300,000 on this project. And we're looking at about a 3 to 4 year cliff for full FDA approval for the device. I'm gonna let you put your hot little hands on it. So this is a prototype. So this is the meter for doctors. That looks about the size of a glucometer, right? Test strips, you can open and look at them if you want. Where are test strips used? In diabetes. So this is a very, very common thing. And thank you to In Vitro Diagnostics for all the amazing science and effort that has gone into this. So I'll start over here. When you get to the end of the table, just pass it to the next table. And then everyone who ends up with it, just leave it on the table over there. Thank you.
Thank you, Dr. Chambers. Thank you very much for inviting us here to talk. And I appreciate everyone who stayed to the last minute to hear me speak, even if they did bribe you with lunch. My name's Bob Mosley. I'm a scientist at In Vitro Diagnostic Solutions, or IVDS, and we've been partnering with the foundation here to develop this point-of-care meter for DADA2 determination in blood. So I just want to give a little bit of background about our company. IVDS was founded in 2017 by Robert Harper, bringing a wealth of experience in bringing point-of-care tests to market in both the medical and veterinary fields, as well as developing novel technologies that kind of serve these sensing solutions that we're trying to create. We're currently fully funded by the NIH, so since 2017, we've raised over $9 million in funding from various grant applications to develop our tests. The main focus of IVDS is novel point-of-care tests for biomarkers for rare diseases, or the type of conditions that we could call underserved conditions, where they really need a point-of-care test, but maybe one does not exist. So we probably have about a dozen tests in various stages of development. Two of our more mature products are the PKU Now, which is an at-home phenylalanine test for PKU patients to manage their lifestyle and interact with care providers remotely. We have another test for G6PD deficiency that we hope to deploy as a rapid screen in malaria endemic regions, and both of those were on track to start clinical studies early next year. Obviously for DADA2, we just recently received, as Dr. Chambers mentioned, our Phase 1 SBIR award from the NIH. So, you know, our proposal was fully peer-reviewed and supported by the NIH, and we're ensuring that the development is under the highest level of scientific rigor and meeting all needed guidelines for this type of test. So really the big advantage of IVDS is the fact that all of our design and manufacturing happens in-house. So we're completely integrated from conceptualization to production. We have the capabilities of making large-scale lot production of test strips in a very iterative manner. So we can do a lot of prototyping and testing, and we can collect and analyze a lot of data in a relatively short period of time. Most of our tests kind of revolve around this reflectance meter that's being passed around that we designed to work with our test strip. Really the point being, our capabilities allow us to be very versatile and very agile and very modular when we're developing our tests, and I think that's why companies like to work with us for development. And I presume this is why IVDS was so attractive to the foundation for this project. So you can kind of see our kit, so to speak, on the left there. Obviously, it's the meter and test strips. We'll include a standard blood draw kit with a finger prick lancet. The test should also work with blood from a venous draw, and it should also work with plasma as well. We do all calculation, results display, data storage, and dissemination through a mobile app. So for one, this allows us to keep the cost of the meter quite low because it doesn't have to do any computation. But really this model we adopted kind of came from our other tests where you either needed to be testing frequently at home and interfacing with a care provider, or in remote regions where maybe you don't have very good access to healthcare, but nonetheless some clinical decision has to be made. Excuse me. In the case of ADA2, we think this kind of model will lend itself really well to long-term longitudinal studies. We understand there's still a lot of uncertainty about how enzyme activity actually relates to disease progression. So we think this type of rapid mobile platform would support studies of that kind really, really well. And we're also really confident that just having this available in clinics or emergency rooms or in offices will get people who are unaware just to start thinking about this diagnosis. And we hope that we can kind of bridge some of the gaps between genotype, phenotype, and activity, and hopefully stop some potential DADA2 patients from falling through the cracks, which I think there is some fear that that's currently happening. Top right, you can see an exploded view of our test strip. It's a cassette housing with a membrane stack. It spreads the sample evenly. The primary and secondary membranes agglomerate and filter out red and white blood cells and other components of the hematocrit. The plasma reaches the reagent membrane at the bottom, and this causes a color change that's proportional to our analyte level, so in this case, the enzyme activity. You can see a little zoom in on the meter. It's got a panel of LEDs that blink under the test strip, and it measures the amount of light that's being reflected back. We tune the wavelength of the LEDs to the absorbance maxima of our indicator, which allows us to get a heightened sensitivity in our tests. And I included just a little snippet here of kind of the raw data that we collect. Quite simply, it's just percent reflectance over time as the test strip changes color. And similar to other enzymatic assays, we're just calculating the rate of the reaction. We're relating it to a known value of activity in the sample or some reference value. And we use that relationship to build our curve set or our calibration equation that we can use to predict activity levels in unknown samples. In the case of ADA, we find that we can get pretty good resolution in as little as 5 minutes. So this is the mechanism that underlies the detection in our assay. So you see we'll start with adenosine as our substrate. And we include an enzymatic cascade, the final step being the oxidation of our indicator, which causes it to change color. Of course, this is in addition to buffering agents and different excipients and bioactive components. We went through pretty thorough testing to try to optimize for the maximum amount of ADA2 activity, obviously to make this test very sensitive and as rapid as possible. We also did a significant amount of testing titrating in EHNA as an ADA1 inhibitor on the strip itself. So as a result, we're confident that we can get in situ attenuation of interference from ADA1, obviously making the test ADA2 specific. So we use ADA2 deficient plasma samples. We spike up with recombinant ADA2, and we dilute in order to make kind of our validation sample set. So we took these samples and we tested them simultaneously on our meter, and we predicted the activity level using our existing curve set. And at the same time, we tested it on our reference assay. At this point, we're using the ADA reagents available from Diazyme. This test has to be modified to make it ADA2 specific. But at least comparing these two assays for the predicted value in units per liter, we see excellent agreement and pretty good precision. On the right, it's a Bland-Altman plot. It's a statistical comparison of two assay methods. So at least in this sample set, we're not detecting any systemic bias from our assay in either the positive or negative direction. And generally, this type of analysis calculates a 95% confidence interval of where your difference range is going to be. And it looks like we're confident that ours can be within about 1.5 units per liter, at least compared to our Diazyme reference. So obviously, we're very excited about our results and progression so far. So this is kind of a tentative development timeline for the ADA2 now. Obviously, we're very grateful for the continued support of the foundation. Additionally, this year we got a state award in New Jersey, and like I said before, we received the SBIR Phase 1 award for continued development. The phase we're in today is putting the finishing touches, so to speak, on our formulation. Really, it involves just optimizing for long-term test strip stability. Once that's finished, we do what we call our design lock. We'll move the formulation under our quality management system. We'll scale up the production of our test strip lots and go through full verification and validation to CLSI guidelines. Next year, we hope to move pretty quickly into preclinical studies. This would probably just involve shipping out a kit to an office, getting some hands-on feedback for usability, opinions on utility of this kind of test. At this point, it would all be de-identified, no clinical decisions. It's more just insight and feedback from the people who would be using the test. From there, obviously, we hope to move into full clinical evaluation and start to move towards FDA submission and approval of this test. So obviously, some of the open questions, the big one is just to continue to work towards consensus. I think what would be really, really helpful for us would be to identify some relevant clinical cutoff values. We expect that the fastest way to get this approved would be for us to submit it as a qualitative screen, maybe plus or minus some relevant activity level to either suggest or rule out further genetic testing. That being said, we are pretty confident about the quantitative abilities of this test. So if there's any non-clinical or analytical labs who wanted to use this kit to kind of support studies involving ADA2 activity, we'd be happy to ship some materials out and collaborate. What we really need are some accurate, validated reference assays. Obviously, we're only as good as our best reference. The modified diazyme reagents are satisfactory for us at this point, but moving forward, we would really like to have a fully validated reference method. And I know there's been a lot of talk about developing different validated methods, so that's really exciting. We would love to work together in that way. At the very least, if there's anyone who has existing biobank samples, specimens with a known activity level that we could use to compare accuracy, I think that would be really helpful. Collaboration, any sites for clinical studies, like I said before, it would be no clinical decisions, no real interface with the patient, just allow you to use this in your office and give us some feedback. But mostly we just need to hear from you, right? So it's very obvious over the past few days that we are not the disease experts. You are all the disease experts. Not to be reductive, but we're just engineers who want to build a platform to spec, and we need you to be able to use it. So we'd love to contact you and reach out and ask some questions. And even patients and families, if they have any feedback on how they manage living with this diagnosis and kind of what they need, these have been really powerful. The relationship we build with patients and caregivers is really powerful for us. So we would love to connect. Okay, if you squint a little bit, I think you'll see my team come into focus. Sorry about that. I've got my email here. Our website is ivd.solutions. I've got cards if anyone's interested and wants to reach out. We would really appreciate that. And anyone who has to travel to get home, I hope you have safe travels. Thank you.